Modern medicine is said to be highly developed, but lifestyle diseases such as heart disease and diabetes continue to increase. And 130 so-called incurable diseases persist. These diseases don't have curative treatment. Existing treatment has focused on the pharmaceutical reduction of symptoms. In recent years, however, attention has been focused on a new treatment. This is regenerative medicine called stem cell therapy. The human body consists of approximately 60 trillion cells. Each cell has a specific role in the body, serving, for example, as muscle or nerve fiber. Certain special cells, however, have no fixed role. They have the potential to transform into any other cell. These are called stem cells. By extracting a body's stem cells, multiplying them in a culture, and administering them to the body, they can transform into healthy cells needed to combat disease. Stem cells are believed to have the ability to repair damaged and diseased cells and tissues, restoring them to their original function. Stem cell therapy is the focus of ongoing clinical studies around the world. Improvement has been shown in the treatment of Parkinson's disease and the paralysis of limbs induced by cerebral hemorrhage. We have been focusing on stem cell therapy's potential. For patients who have given up on conventional treatment, who have given up hope, we are moving forward with new technology. Stem cell therapy must follow the guidelines of the Ministry of Health, Labor and Welfare. Special care must be taken, particularly with the following 3 items: explaining scientific evidence shown from basic experiments or clinical trials, creating safe and secure procedures, a thoroughly informed consent process. To ensure objectivity, universities and research institutions must set up third-party ethics committees. Approval must be obtained for the safety and efficacy of medical activities. Though we could have opted for fewer restrictions under the Japanese Medical Practitioners Act, we voluntarily adopted the Ministry of Health guidelines. Having set up a comprehensive system, we started cutting-edge stem cell therapy in 2011. The Cell Processing Center, the core of our treatment, is the largest in Japan and equipped with the latest technology. Our sterile operating room has a cleanliness of class 100,000. A hygienic environment is a constant consideration. Thoroughly informed consent is essential to conducting the treatment with respect for the patient's will. It is likely that many patients will have questions and fears about any new health procedure. Our doctors candidly disclose the information as much as possible to answer such questions and fears. We elected to use adult stem cells harvested from subcutaneous fat for our treatment. Here, let us compare the benefits of our procedure with others utilizing other stem cells. Current stem cell research approved for clinical regenerative medicine can be generally divided into 3 types. Those using embryonic stem cells, ES, harvested from an early stage of fertilized human embryo, induced pluripotent stem cells (iPS) created by incorporating genes into human skin cells, somatic stem cells harvested from various tissues in the body. Though ES and iPS cells can transform into any cell in the human body, many challenges remain to be overcome before clinical treatment can be made effective. Somatic stem cells taken from the patient's own body, are not subject to immunological rejection. Recent studies have proved that stem cells derived from muscle, fat, and bone marrow can be successfully transformed into bone, blood vessels, and nerve cells. Stem cells made from subcutaneous fat harvested from a patient's own body provide relief to patients recovering from bone marrow treatment without fear of infection. We have made real achievements in over 400 cases using adipose tissue-derived stem cells. Rigorous operational management ensures that our cell culture facilities conform to good manufacturing practice, GMP standards with the high levels of safety and quality management required of pharmaceutical manufacturers. In addition, we have created a secure cell culture medium. To improve efficiency, cells are usually cultured in a medium derived from animals, such as fetal bovine or pigs. We do not use animal-derived culture mediums. This eliminates the risk of infection or allergic reactions. Our proprietary culture technology has, moreover, achieved high levels of efficiency. Combining safety with technological innovation, we are poised for success. Stem cell therapy is a new field of healthcare medicine. Continued study is required to make safe, reliable, and effective treatments. We believe this requires us to build strong relationships between patients and doctors. We continue to collaborate on cutting-edge medical technology with national laboratories and universities. Contributing to society with our technology, we are always thinking about the future of our patients and will continue on with our efforts. This corner is for sharing important notes about the use of adipose tissue-derived stem cells. The first step in starting a patient's stem cell therapy is counseling. We provide accurate information and discuss the treatment thoroughly. The second step is a blood test to check for any other viral, pathogen, and microbe infections. Test results are available in approximately 1 week. If all results are negative, we can make the decision to proceed with fat cell harvesting. About 5 to 10 grams of subcutaneous fat cells are harvested mainly from the abdominal tissue. We can harvest fat cells from the gluteal region of patients with insufficient abdominal fat. After making a 2 cm incision, fat cells are directly harvested with a syringe. The incision may leave a scar, but reduces the length of the operation and puts less of a burden on the body. Stem cells harvested from fat tissue are cultured over 4 to 6 weeks. Strict quality control ensures utmost care is taken with stem cell cultures. Sterile conditions for stem cell cultures are ensured with endotoxin, mycoplasma, and other tests. Cultured stem cells are infused to the affected area or administered by injection. A reserve supply of cultured stem cells are frozen for the future. Regular follow-up examinations are conducted to measure efficacy and to monitor the patient's long-term health.